Hearty greetings. I am Mrs. J. Albinovins from Holy Cross College Autonomous Nagarkoil, presenting about the potential source of nutraceuticals from marine flora and fauna. Introduction. As we all know, approximately 70% of our planet's surface is covered by oceans. Marine biodiversity makes a potential source of maximal amounts and varieties of novel bioactive compounds. These compounds are mostly synthesized as secondary metabolites by the marine microflora and fauna. Actually, large number of potential bioactive compounds are found in the oceans as well as deep sea with biological properties and pharmaceutical efficacies. They are used in the treatment of malignancy, anemic disorders, dysentery and diarrhea, obesity, diabetes, dermatitis, etc. Natural products of marine are mainly accumulated in invertebrates such as sponges, tunicates, bryozoans and mollusks. Approximately more than 2,400 natural products have been isolated from macroalgae. Seaweeds have a great potency against diabetes, microbial infections and inflammations because they are a great reservoir of many chemical constituents like acids, alkaloids, amines, antibacterial substances, antifungal substances, antiviral substances, lipids, sterols, steroids, fatty acids, phenolic compounds, phytohormones, pigments, proteins, peptides, amino acids, sugar, alcohols and vitamins. Marine bacteria. Marine bacteria are prolific producers of valuable secondary metabolites. The marine isolate Pseudomonas is not well explored and only a limited number have been reported as producers of bioactive compounds. A bacterium strain KMM3042 produce some bioactive substances such as pyrols, pseudopeptide, phenazine, benzaldehyde, quinolone, phenanthrene, phthalate, andromide, moiramides, safran and bushrin. Some of these bioactive compounds are antimicrobial agents. Stenotrophomonas strains showed remarkable antimicrobial and antifungal inhibitory activity. Stenotrophomonas maltophilia possess biocontrolling capabilities. Anadara broughtoni exhibit strong antimicrobial hemolytic properties. Pseudoaltromonas inhibit methicillin-resistant Staphylococcus aureus strains due to the 3,5-tetrabromo-2,2 diphenyldiol. Pseudoaltromonas luteoviolica inhibit the growth of protist. Pseudoaltromonas tunicate produce a yellow pigment that possesses antifungal activity. Marine fungi. Marine fungi have been known to produce a wide variety of bioactive metabolites that possess anti-cancer, antibacterial, antiplasmodial, anti-inflammatory and antiviral activity. A novel anthraquinone derivative with naphtho123D Chromino 27 dione skeleton was isolated from a marine filamentous fungus Aspergillus glaucus. It was found to exhibit cytotoxic activity against the cell lines. Penicillium species isolated was found to exhibit antitumor activities in their alkaloid-rich extract due to the presence of meleagrin D and E and roquefortine H and I. Ambylomyces species possess potent antimicrobial and antifouling compounds. Cladosporium species exhibited antibiotic and antifouling activity. Fusarium species possess novel antimitotic and antifungal activity from its fuserlin E. Pseudella escherichia produce a novel antibacterial dioxopiperaxin dehydroxy bisdithiobis with potent antibacterial activity against the methicillin-resistant, multi-drug-resistant Staphylococcus. A marine fungus, Exophialia, exhibit antibacterial activity due to the presence of two novel antibacterial aspirone derivatives, that is chlorohydroaspirone A and B. The marine fungi, Lachnum, Papyraceum, exhibit nematicidal effect. Poma herbarum strain has residual scavenging activity against DPPH. Marine actinomycetes. Actinomycetes are well known to be the producers of secondary metabolites. Many well-known antibiotics such as streptomycin, erythromycin and tetracyclines were produced by them. They are producers of novel anti-tumor, anti-malarial and antimicrobial compounds. Nocardiopsis lucentis produced 4 novel 3-methyl-4-ethylene containing the peptides called as lucentamycins A, B, C and D. Lucentamycin A and B exhibited strong in vitro cytotoxicity against the human colon carcinoma. Streptomyces species produce mansoramycin A, B, C and D and the known 3-methyl-7-methylamino 5,8-isoquinoline. These compounds exhibit strong cytotoxicity against lung cancer, breast cancer, melanoma and prostate cancer drugs. Macrodiolide Tartolon D from Streptomyces species exhibits cytotoxic activity against the human tumor cell lines of lung, colon and breast. Saccharomyces species yielded a novel alkaloid, lodopyridone, with cytotoxic to human colon cancer cells. Marinispora has chlorinated basindole pyrols and linamycins A to E that exhibited a broad spectrum antimicrobial activity. Microalgae, cyanobacteria, also known as blue-green algae, produce secondary metabolites with antifungal, antiviral, antibiotic and other properties. They exhibit selective bioactivity against the vertebrates, invertebrates, plants, microalgae, fungi, bacteria, viruses and even the cell lines. Thus they possess a great pharmaceutical value. Eulythia cyclamide and patilamide A belong to cyanobactins produced by the cyanobacteria that has a potent antimicrobial, that is antimalarial, anti-tumor and multi-drug reversing activities. Viridamide A is produced by the blackish green mat forming filamentous cyanobacterium. Oscillatoria nigroviridis showed anti-trypsinomal and anti-leishmanial activity. Cladophora rupestris, Codium fragile, Alva intestinalis, Alva latuca were found to exhibit antiprotozoal activity. Eicosapentaenoic acid were produced from the marine diatom that showed activity against the range of both gram-positive as well as gram-negative bacteria. They are used as nutraceuticals. Microalgae are rich sources of most of the nutrients such as beta-carotene, vitamin C, A, E, H, B1, B2, B6 and B12, astaxanthin, polysaccharides and polyunsaturated fatty acids. Thus, their bioactive molecules are produced commercially for use as food additives, infant milk formulations and dietary supplements. Macroalgae. Macroalgae are commonly known as seaweeds. They are the most popular algae in the nutraceutical industry and are used in a great variety of food and food ingredients, especially in Asian countries. Macroalgae are also well known for the production of agarose. They are also an important source of many bioactive metabolites and natural products that possess many nutritional and therapeutic functions. Some of the examples of bioactive constituents are proteins, furanones, polyunsaturated fatty acids, kynic acid, phenolics, pigments, fluorotannins, picocolloids, and minerals. Likewise, red and brown seaweeds are also good sources of many vitamins, minerals, proteins, and essential fatty acids. They are also used to prepare bioactive peptides and to improve protein digestibility. Further, antihypertensive bioactive peptides were also isolated from the macroalgae. They can act as angiotensin converting enzyme inhibitors. Coral reefs. Corals are stationary animals and have evolved chemical defenses to protect themselves from the predators. Coral reefs are sometimes considered the medicinal cabinets of 21st century. Coral reef plants and animals are important sources of new medicines being developed to treat cancer, arthritis, human bacterial infections, Alzheimer's disease, heart disease, viruses and other diseases. In the future, coral reef ecosystem could represent an increasingly important source of medicinal treatment, nutritional supplements, pesticides, cosmetics, and even other commercial products. Sponges. Sponges are considered as the chemical factory in marine environment because of its immense production of chemically diverse compounds. These compounds possess remarkable bioactivities. This great potential has aroused applications of sponge-derived compounds as therapeutics, and at present a number of promising compounds are in clinical and preclinical trials. Recently, nutraceuticals have received considerable interest among the health conscious community because of its multiple therapeutic effects. Sea grasses. Marine plants are also known to produce a large number of structurally diverse secondary metabolites. They are found to be economically important in the field of food additives, nutraceutical as well as drugs. The phytochemical present in the seagrass exhibit antibacterial, antioxidant, anti-tumor activity. The novel chemical entities are the source of treatment for many diseases like cancer, AIDS, inflammatory conditions, arthritis, malaria, and large variety of viral, bacterial, and fungal diseases. Several species of seagrasses are used as human food or as raw material for the production of compounds of nutritional interest. Marine animals. Marine fish species and algae have been identified as the sources of polyunsaturated fatty acids which are rich in omega-3 or omega-6 fatty acids. The presence of these unsaturated fatty acids in marine derived foods increases their applicability as nutraceuticals in the food industry. Thank you. Ocean is the world of pharma and world of nutrients. Thank you one and all.